So sarcomatoid and rhabdoid renal cell carcinoma is an aggressive form of RCC that occurs in up to 20 to 25% of patients with metastatic renal cell carcinoma. This form of disease is overrepresented in patients presenting with metastatic disease and is associated with a poor prognosis historically. Patients with the target therapy era, we had much better options for patients with clear cell RCC, but unfortunately patients that had a sarcomatoid or rhabdoid features responded very poorly to these target therapies. I should say that these sarcomatoid and rhabdoid features occur on both a background of clear cell RCC, but also a background of non-clear cell RCC. So very luckily, in the recent large randomized controlled trials of immune checkpoint inhibitor-based agents for renal cell carcinoma, the subgroups of patients with sarcomatoid RCC seem to be responding very well to immune checkpoint inhibitors, such as in the nivolumab ipilimumab trial, the pembrolizumab axitinib trial, and the avelumab axitinib trial, but also the atezolizumab bevacizumab trial. All these subgroups, relatively small, 50 to 60 patients, showed that there was a benefit to immune checkpoint inhibitors in patients with this disease. Now, what we set out to do is to ask this question at a much larger scale to confirm whether this benefit was in the sarcomatoid but also in the rhabdoid group of patients, and whether it was confirmed in monotherapy immune checkpoint inhibitors, but also combination immune checkpoint inhibitors. We asked this question in 3 cohorts, the first one being a Harvard cohort from Dana-Farber, MGH, BWH, and BIDMC, with more than 200 patients. We found that benefit to immune checkpoint inhibitors in patients with sarcomatoid and rhabdoid RCC. We confirmed these results in the IMDC dataset, which is a large dataset of patients with metastatic renal cell carcinoma, including more than 900 patients with sarcomatoid and rhabdoid RCC from more than 40 institutions across the world. We then confirmed that also in subgroup analyses of 2 clinical trials, the CheckMate 025 and CheckMate 010 clinical trials of single-agent nivolumab. We also found an overall survival benefit in that pooled analysis. So then we set out to ask, what is underlying both the poor prognosis of these patients? Are there molecular features that explain them, especially in the target therapy era? And also, why are these patients responding to immune checkpoint inhibitors? At the DNA level, and this is a pooled analysis from multiple cohorts, we found that tumors that have sarcomatoid and rhabdoid features seem to be enriched in mutations in NF2 and BAP1, in deletions at the CDKN2A locus, and in amplifications in the EZH2 locus, which would probably explain the poor prognosis, because mutations in particular in BAP1 and even NF2 have been associated with a poor prognosis in RCC. Then at the RNA level, we found that there was a MYC-related signature that was upregulated in these tumors, and also the cell proliferation signature seemed to be upregulated, suggesting that this may be underlying also the poor prognosis in these tumors. We also found that these tumors have a very immune-inflamed phenotype at the transcription level, with pathways such as IL-6, JAK-STAT3, and IL-2 STAT5 signaling increased, but also infiltration by CD8 T cells and high expression of PD-L1 on the tumor cells of these tumors. So in conclusion, I think that sarcomatoid and rhabdoid RCC is historically associated with a poor prognosis. We tried to explain in this study what are the molecular features underlying the poor prognosis of these tumors. We believe it's a combination of DNA and RNA alterations, but also these tumors are now responding very well to immune checkpoint inhibitors. We provide the largest data sets to date showing that, but we also try to get at the mechanisms underlying this improved responsiveness to immune checkpoint inhibitors using a combination of RNA analysis, immunofluorescence, and IHC to get at that question, and finding that these tumors have a very immune-inflamed phenotype.